Aloha everyone, and welcome to our lecture today on Chapter 3. We will be talking all about our amazing cells, their structure and function. Of course, we have, first of all, our learning objectives for today. So by the end of this lecture, you should be able to, first of all, describe the cell theory. Secondly, be able to compare and contrast prokaryotic and eukaryotic cell structure and function. Third, be able to describe our cell membrane structure. Fourth, explain how solutes or molecules are able to cross that cell membrane using 4 different transport mechanisms. And then the last learning objective has to do with kind of the background story in Chapter 3, where it talks a lot about antibiotics. So I want you to be able to understand how antibiotics are able to kill bacteria cells without harming human cells. So let's go ahead and get started. Our first learning objective is to learn about the cell theory. I briefly mentioned cells and the cell theory in our Chapter 2 lecture. So the word cell was actually first used by a scientist known as Robert Hooke back in 1665, and he was actually looking at cork tissue under a microscope, and he saw what appeared to be these small little chambers, and he termed them cells. The cell theory came about later in 1838, and it was first proposed by 2 different German scientists, Schwann and Schleiden, and they coined this cell theory. The cell theory states that it kind of states 3 different things. First of all, the cell is a basic unit of life. You cannot have life without cells. Secondly, all living things are made up of cells. These two kind of go together. And then third, every new cell doesn't just appear out of thin air. Every new cell must come from a pre-existing cell. So these 3 components are what we term the cell theory. And even though all cells, cells are the basic unit of life, not all cells are alike, right? Cells come in many different shapes, many different sizes, and they're able to perform many different functions depending on where they are found. The second objective for today's lecture is to be able to distinguish between what we term prokaryotic cells and eukaryotic cells. So prokaryotic cells are cells which lack internal membrane-bound organelles. So this is going to be mostly, think of bacteria cells, right? So your prokaryotic cells are going to have a cell wall around them. They're going to have a cell membrane. They're going to have their DNA or their genetic material just kind of floating around in that liquid cytoplasm. Whereas for a eukaryotic cell, so U means true, so think of like true cells. This is going to be the type of cells that we have. They're going to have membrane-bound organelles, including the central nucleus that's going to house that genetic material. Looking a little more at our prokaryotic cells, they are going to lack a nucleus, and they have an absence of most organelles in the cell. The cells are going to contain kind of this circular ring of DNA. The cell wall is normally going to be present, so you can see the cell wall right here. They're going to be unicellular, meaning single-celled. And so again, you're going to see this DNA just kind of floating around in the cytoplasm. There are no organelles, again, right? Nothing that's membrane bound. And they have a cell wall. The cell wall, again, right here is actually made of peptidoglycan. And we'll talk more about this when we look at how antibiotics work. But the cell wall is just kind of this rigid structure enclosing this plasma or cell membrane. So again, think kind of bacteria cells when you think of prokaryotic cells, at least in terms of this class. Then we have our eukaryotic cells. These are cells with DNA enclosed inside of the nucleus. So eukaryotic organisms are going to have organelles. Organelles are membrane-bound compartments found in eukaryotic cells that are going to carry out very specific functions. So eukaryotic cells possess many specialized organelles, which we're going to talk about in the following slides. We're going to go through just some of these organelles. Eukaryotic organisms can be either unicellular or multicellular. In terms of this class, we're mostly going to be thinking of multicellular eukaryotic organisms. And like I said, we're going to talk about some of these really unique organelles that we find inside eukaryotic cells, meaning when I say unique, meaning they have unique functions. So we'll talk about our nucleus, we'll talk about the Golgi apparatus, we'll talk about the mitochondria, ribosomes, and endoplasmic reticulum in the following slides. The first organelle we're going to look at is the cell's nucleus. So the nucleus encloses the cell's DNA, and it is responsible for kind of housing the DNA in there and kind of responding to different things that could take place inside of the nucleus. So it's going to interpret the genetic instructions that take place there, and it is surrounded by the nuclear envelope. So you can see here your nucleus in the center of that cell, right? Kind of looks like this little circular globe. If we zoom in on that, you have your nuclear envelope on the outside. It's kind of a membrane around it. And then inside of that is going to be your genetic material. You have your nucleolus inside of there as well. But the main thing I kind of want you to know about the nucleus in terms of this class is that inside of it you are going to find your genetic material. So you can kind of think of that nucleus as being really important because it's keeping that genetic material kind of contained instead of just floating free in the cytoplasm like we see with prokaryotic cells. The next organelle is the endoplasmic reticulum, which sounds a little funny. A lot of times it's referred to as the ER. And this is just kind of this network of membrane-bound almost sacs that kind of connect to one another. So you can see here that endoplasmic reticulum. It looks like little sacs, little folds that connect to one another, almost like pipes, right? There are two different types of endoplasmic reticulum, and that's because in the endoplasmic reticulum, we have both proteins and lipids being synthesized, or in other words, being made in the endoplasmic reticulum. So the rough ER is going to be studded with these little ribosomes in blue here. So this is your rough ER. The ribosomes are necessary to make proteins. So that is your rough ER, that's where you're going to make the proteins. Whereas your smooth ER over here, you can see it doesn't have those ribosomes. That's because the smooth ER is where we're going to make lipids. Next, we have the Golgi apparatus, another kind of funny sounding word. These are your stacked membranous discs. This is going to receive proteins from the ER. So a lot of times you kind of see how the Golgi apparatus is going to be near or kind of connected to your ER. So here's your ER, that endoplasmic reticulum, and then right next to that you have your Golgi apparatus. So it receives protein from the endoplasmic reticulum via these little vesicles. They're kind of these little circles, right? So imagine your ER is right here. Then you have your proteins inside of these vesicles going to your Golgi apparatus. And the function of that Golgi apparatus is actually to package and transport those proteins. So these vesicles are delivering those proteins to the Golgi apparatus. As they make their way through this Golgi apparatus, They're going to be processed to complete their structure and kind of give them a purpose to be used in the cell inside of the human body. Next, we have the mitochondria. You probably heard mitochondria being referred to as the cell's powerhouse before, because the function of mitochondria is to actually extract energy from the food that you eat. So it converts energy into a useful form. So the mitochondria, if you look at the structure, it also is double membrane bound. So you have this inner membrane here, you have an outer membrane here, and then you have inside of here where you're going to be taking and extracting the energy from the food that you eat. So again, think of mitochondria as being the cell's powerhouse. A cell organelle that you might not be quite as familiar with is your lysozymes. These are actually chock full of digestive enzymes. So they're going to be enzymes that help you to break down things. So they're going to break down worn out cell parts, food molecules. This is an example here. It shows you a lysosome right here. So it shows you here outside of the cell, these materials will come in, they'll go inside of that lysosome, and that lysosome is able to kind of break down these structures because they have all those enzymes inside. The last portion of the cell organelle that I'm going to talk a little bit about is the cytoskeleton. So this is essentially a network of different protein fibers, and its functions are many. So it includes helping to support the cell. It can be used for the motion of the cell when we talk about flagella, which are kind of the little tails that you'll see on cells that help the cell to move. Think of, as example, human sperm that has a flagella on the end of it. And it also allows movement of structures within the cells too. So we're not going to talk too much about it in regards to this class, but if you were to take, say, a physiology course, you would learn about the different types of proteins found in the cytoskeleton. So you can have different types such as your microfilaments, which are going to be pretty small. You have your intermediate filaments, which are intermediate or medium in size, and then your larger microtubules. And it just kind of shows you here, these microfilaments are kind of right inside that cell membrane. You have your intermediate filaments that kind of branch throughout, and then your microtubules are found throughout that cell as well. So just to kind of recap a little bit here, when we look at our prokaryotic versus our eukaryotic cells, what we find is that there are actually 4 structures that are found in both prokaryotic as well as eukaryotic cells. So they each are going to have a cell membrane, which is going to kind of keep everything together inside of that cell. They both are going to have cytoplasm. Cytoplasm is just kind of a gelatinous, kind of aqueous material that's inside of that cell. Kind of floats around inside, and all the organelles, or in the case of your prokaryotic cells that don't have organelles, just all the structures are kind of floating around inside of that cytoplasm. Both have ribosomes. Ribosomes are kind of this complex of RNA and a protein that carry out the process of protein synthesis. So you can see prokaryotic cells have these, in purple, these ribosomes, as well as eukaryotic cells. We talked about ribosomes are in the rough ER of eukaryotic cells, right? And then we also find that prokaryotic cells and eukaryotic cells both have DNA, which is that genetic material. In the case of prokaryotic cells, that DNA just floats around in the cytoplasm. In the case of eukaryotic cells, that DNA is housed inside of the nucleus. And your book doesn't talk too much about this, but I just wanted to kind of show you the difference as well. When we talk about eukaryotic cells, both animals and plants are eukaryotic, but there are differences between animal and plant cells as well. And there are 3 major differences. If you look at these 2 pictures, this being an animal cell, this being a plant cell, you might kind of notice the three differences. So look for a second, see if you kind of pick up what they are. The first one that I think students usually notice is this huge vacuole right here in the plant cell. So that vacuole is used for storage of water and nutrients. Another difference is this very rigid structure around the outside of a plant cell. That is a cell wall. You do not find a cell wall on an animal cell. And then the third difference is chloroplasts. So in green here, you see this chloroplast. Plants and algae have chloroplasts. This is the site of photosynthesis. And again, we'll be talking about photosynthesis later in this course. And photosynthesis, these chloroplasts are used to help to capture that sunlight energy and convert it into a usable form. So those are the three major differences between your animal and your plant cell. It's that plant cells have three things animal cells do not. They have chloroplasts, cell walls, and vacuoles. Next, we're going to move on to our third learning objective, which is to learn about the structure of the cell membrane. All cells have a cell membrane, and this cell membrane is what we term semi-permeable, meaning some things can pass through, some things cannot pass through. The cell membrane is often referred to kind of as being, you may have heard of the fluid mosaic model. And essentially what this means is that that cell membrane is composed of many different pieces. So this model explains kind of various observations showing the structure of functional cell membranes. These can kind of float in and through, making it kind of like a fluid mosaic. A mosaic is a picture or a pattern that's created by arranging all these little small pieces together. It has many different parts, and that's kind of why we think of our cell membrane as being a mosaic. Right? There's many different parts to it. There's the phospholipids that we'll talk about. There is different types of fats, such as this shows you cholesterol. There are different proteins that form kind of channels in the cell membrane. So again, things are kind of moving through the cell membrane, making it fluid. So that's why we term the cell membrane the fluid mosaic model. Looking a little more at the cell membrane structure, it is a selectively permeable phospholipid bilayer. So the structure of the plasma membrane allows some substances to easily cross it, but it prevents other substances from crossing. It's kind of like if you think of a bouncer at the club, right? It lets some people in, but not everyone. Or if you want to take it away from the example of the club, think of it as like a screen door. Right? Or say a certain size dog door. Lets some things in, doesn't let other sized animals in. So both layers of the plasma membrane have hydrophilic heads of the phospholipids kind of pointing towards the liquid on the inside and outside. So to make that make sense, I need to talk to you a little bit about phospholipids. These are the primary component of your cell membrane. And a phospholipid structure, you can see it here, they have a head region that is hydrophobic. We talked about this word in chapter 2. Hydro means water, phobic means fearing. So these are water fearing. They do not like to be around water. Then, sorry, and I messed that up. It is your hydrophobic tails right down here. So nonpolar hydrophobic tails. Then you have your hydrophilic or water-loving polar heads. So I apologize for misspeaking a little bit ago. Your head region is hydrophilic. Your tail region is hydrophobic. So these are going to be arranged in a certain way on the membrane that makes them essentially happy. So if you look at it, what happens is these hydrophilic heads, meaning they like water, you're going to see them on the outside, so they're going to be next to the extracellular fluid. You also see them facing the inside of the cell for that intracellular fluid or the cytoplasm, right? So the head region of the phospholipid is by water because it's hydrophilic, it likes water. Whereas the tails orient towards each other. So they are on the inside portion here not exposed to any water because the tails are hydrophobic or water-fearing. So because cells reside in a watery solution, that extracellular fluid that you see on the outside here, and they contain a watery solution inside of them, this cytoplasm, the plasma membrane essentially forms a circle around each cell so that these water-loving heads, again, are in contact with the fluid, and the water-fearing tails are protected on the inside. So that's a big thing in terms of the structure of the cell membrane. So in terms of this class, we're not going to talk too terribly much about all the other components of the cell membrane. But I did put a figure over here just to kind of show you that it's much more complicated than just this phospholipid bilayer, right? You have these large things in purple here. These are your different channels formed by different types of proteins. You also are going to have these sugar molecules attached to the cell membrane. You also have cholesterol in the cell membrane, which is a fat. So you have multiple different things in this cell membrane, which again is why it's called a mosaic. Again, for the purposes of this class, mainly focusing on this phospholipid bilayer. The hydrophilic or water-loving heads, hydrophobic water-fearing tails of that phospholipid. They orient, so those tails are on the inside here, or they're facing each other, no water there. The heads face the cytoplasm, the heads face that extracellular fluid because they like water. So again, your cell membrane is what we term semi-permeable. So it's going to allow some substances to easily cross it, prevent some substances from crossing it on their own. They might need another molecule to help them cross it, or they might need energy in order to cross it. And that kind of brings us to talking a little bit about our next learning objective, which is how exactly are things transported across that cell membrane. So we have two different types or main broad categories of transport. So things can either pass through passively, meaning no energy is required, and this is what we term passive transport, or we have active transport, meaning we must have energy in order for that molecule to to be transported across the cell membrane. So in the next few slides, we're going to talk more about these transport mechanisms and the different types of passive transport, and then we'll also look at active transport as well. We're going to begin by looking at passive transport, which again requires no energy input. There are three different types of passive transport we're going to talk about. We have simple diffusion, osmosis, and facilitated diffusion. Something I want you to keep in mind is that all passive transport involves a natural movement of the molecules going from high concentration to low concentration. In other words, they're moving down the concentration gradient. So it doesn't matter which of these three types of passive transport we are talking about. They are all moving down that concentration gradient, which is why no energy is required. So always going from a high concentration to a low concentration because we're trying to equal things out and get it equal on each side of that membrane. The first type of passive transport that we're going to talk about is simple diffusion. This is the movement of very small hydrophobic, meaning water-fearing molecules across the membrane from an area of higher concentration to an area of lower concentration. So molecules have a natural drive to diffuse down concentration gradients that exist between the outside of the cell and the inside of the cell. Molecules that are able to passively diffuse through the membrane include lipid-soluble substances, very small substances that can kind of pass through that membrane easily. So again, this is simple diffusion. We're always going from high to low concentration. And that's illustrated over here. So you have your solutes in red, and you have a high concentration of that here outside of the cell. Then you have your plasma membrane, and you have a much lower concentration of those solutes inside the cell. So it's going to naturally have this flow from high to low concentration through that cell membrane, and that is simple diffusion. Next, we have the passive transport of osmosis. Osmosis is very similar to simple diffusion. The only real difference is that osmosis is not the diffusion of solutes. Osmosis is the diffusion of water across that semi-permeable membrane. So water is also going to want to kind of flow in a way to equal out that concentration gradient. So it's going to want to flow in a way that the solutes are equal on each side. So if you have more solutes somewhere, water is going to want to flow towards that to lead to there being more water there, kind of decreasing that concentration, equaling out the concentrations. So it's kind of thinking it in the opposite light of simple diffusion, right? Because now we're talking about fluid moving, that water moving, versus the solutes moving. So we can have an isotonic solution. So isotonic is where that solute concentration is the same as the cell's cytoplasm. So in an isotonic solution, water, there's going to be no net movement of water into or out of the cell. It's going to flow in and out at the same rate. And these are just red blood cells shown here for illustration, right? So isotonic, that's kind of what we want inside of our bodies, right? We want our red blood cells to remain intact and look just like this. So we want to have them be in this isotonic environment, water in and out at the same rate. If we have a hypertonic solution, so cells that are placed in a higher solute or hypertonic solution, they will tend to lose water and shrivel because this solution out here, water is going to want to flow out and even that out, right? It's going to want to decrease that solute concentration that's in this hyper, meaning more. So there's more solutes out here. Water's going to want to flow out and try to even that out, right? So that's going to cause these cells to shrivel up. If we have the opposite situation, so hypotonic, meaning there is less solute, lower solute or hypotonic solution, then the cells will tend to take up water because we want to make it so that this is outside solution is equal to what's inside of the cell. So in order to do that, we'll have the cell take up water to make it so that the cell has less solute to try to even out with a hypotonic solution. So these cells are going to take up that water and get so bad that as they swell, they get too big and they lyse, which means to break because they are too full of water. So again, what we want is this isotonic solution, because in all cases, water moves in a direction that will tend to even out the solute concentrations on each side of the membrane. So again, here in hypertonic, we have high solutes out here, right? So water is going to tend to flow out to even that out. Whereas in hypotonic, we have less solute out here. Water is going to flow in to try to make it so the cell has less solute and is equal to that the medium that it is in, that hypotonic solution. So all cases, water flows in a direction to even out the concentration on each side of that membrane. The last type of passive transport that we're going to talk about is facilitated diffusion. This is where large or hydrophilic water-loving molecules are able to cross the membrane. They cannot do this by simple diffusion because they are too large or because they're hydrophilic, right? So simple diffusion isn't an option, but they are able to do facilitated diffusion because they can cross the membrane by using the assistance subunits of what we call transport proteins. These transport proteins, you can see those over here in green, they are going to act as either a channel, they're going to be a carrier, or they're going to be a pump to help molecules cross that cell membrane. So they're able to move specific substances across the cell membrane without requiring any input of energy. So if you have these larger red molecules that cannot pass by simple diffusion, they're going to use these green transport proteins. You can see they're forming this nice little channel in the cell membrane, and that allows these larger solutes to cross through into the cell membrane. So that is facilitated transport. Facilitated means you need the help of these transport proteins. The last type of transport that I want to talk about is not passive transport, but rather active transport. So like facilitated diffusion, it's going to use the help of transport proteins, but those transport proteins just aren't enough. So unlike facilitated diffusion, active transport is actually going to require an energy input, and it's going to pump the solutes to the side of the membrane with the higher solute concentration. So remember, passive transport always went from high to low concentration, right? Active transport does the exact opposite. Here you're going from low to high concentration. So because you're going against the concentration gradient, that is exactly why you're going to need that input of energy. So over here, you can see we have low solute concentration, but if we add in energy, we're able to take those solutes, move them through this transport protein channel, and go up to the higher solute concentration. So in this case, we're moving up that concentration gradient. So that's big differences between active transport and facilitated diffusion are that, first of all, we need energy. Secondly, we're going against the concentration gradient. Okay, so finally we're on to our last learning objective, which is to learn exactly how antibiotics work. How do they kill off bacteria cells without hurting human cells? And that kind of goes along with the whole story of the chapter. I love how your e-text kind of weaves these stories into, it has a theme for each one of your chapters. So I kind of wanted to end this lecture by talking about how antibiotics work. Back in 1928, Alexander Fleming actually discovered that the fungus Penicillium notum was actually capable of killing many kinds of bacteria. And here you can see his kind of original like petri dish here. Here's this mold, and then around that mold, which is a type of fungus, there was no bacteria. And he thought, that's kind of odd. And he did a lot more experiments and found that yes, this was actually the case. Something about that fungus killed bacteria. It killed many different types of bacteria. So this kind of led to the first antibiotic being produced, which was penicillin. Antibiotics are preferentially going to kill bacteria without harming the human or animal host. And they target what's really unique about bacterial cells. And that thing that is unique is that bacteria cell walls are rigid, meaning they're strong, due to their composition. They are composed of peptidoglycan. Peptidoglycan is a polymer, so that means it's many monomers put together, made of sugars and amino acids. So both carbohydrates and proteins together. So kind of makes sense in terms of this word, right? Peptido, protein, glycan, sugars. And this cell wall allows bacteria to survive in watery environments. So in other words, it makes it nice and strong, keeps those bacteria cells intact. So antibiotics are going to target this specific cell wall formed with peptidoglycan because it is unique to bacteria cells. We don't have cell walls, right? We definitely don't have cell walls made of peptidoglycan. So how exactly does this work? Well, penicillin, and other antibiotics as well, are going to weaken the peptidoglycan cell walls. when that antibiotic weakens the cell wall, what that means is it starts to break it down. And that cell wall being rigid is what allows the bacteria cells to live inside a watery environment. So when that cell wall starts to break down, those bacteria cells are going to fill up with the water from the environment that they're in. And as we talked about, as cells start to swell, there comes a point where they just can't swell anymore. And what happens is they lyse or they burst due to that process of osmosis that we talked about. So that's kind of shown here, right? So in the presence of penicillin, that cell wall is going to be weakened, water flows in, and eventually that cell is just going to rupture due to having too much water flow into it. Bacteria obviously are not the only organisms with a cell wall. So we talked about plants have cell walls, fungi also have them. But bacteria are the only ones that have a cell wall made of peptidoglycan. And that is why penicillin is such a selective bacteria killer. So to kind of take you back in history a little bit, a lot of soldiers and civilians had died in World War I, not necessarily just from direct hits in battle, but more so from infected wounds. And there were few kind of antibacterial medicines available. When we heard about penicillin, it became the focus of research during World War II because we're really concerned going into another world war that soon after World War I because we're still trying to deal with all the casualties from World War I, right? So in 1941, Oxford scientists approached the US government and they actually asked for help in growing penicillin on a large scale. And in the US, by using a byproduct of our large-scale corn processing as kind of a culture to grow the penicillin in, we were able to grow that fungus and produce penicillin in much greater quantities. The wide-scale manufacture of penicillin began in the 1940s, and it became kind of the new wonder drug during World War II. Something to kind of point out, though, is that penicillin does not work on all bacteria. And that's because we have two main types of prokaryotic cells, or two main types of your bacteria. We have both gram-positive and gram-negative. So gram-positive bacteria, which is what penicillin is able to target, is bacteria that has a peptidoglycan cell wall, and when it is stained with a crystal violet dye, it's actually going to turn purple, right? So gram-positive bacteria have very thick cell walls that retain the dye, making them appear purple or blue underneath the microscope, or even on your kind of petri dishes too. So gram-negative bacteria, on the other hand, has a thin cell wall, and it's not going to retain that blue dye. And it makes them appear kind of pink or red. So gram-positive bacteria is going to have multiple layers of that peptidoglycan, while your gram-negative bacteria has thin peptidoglycan cell wall bacteria, or a thin peptidoglycan cell wall surrounded by kind of this outer membrane. So this gram-negative bacteria, it prevents penicillin from actually reaching the peptidoglycan cell wall underneath because that cell wall is surrounded by that outer membrane. So some examples of gram-positive bacteria would be strep, staph, and some other listeria species, whereas gram-negative bacteria can cause some serious infections such as cholera, E. coli, and salmonella. So penicillin is really useful for your gram-positive bacteria. But again, because your gram-negative bacteria has this membrane surrounding that cell wall that penicillin targets, it's not effective on your gram-negative bacteria. So even though penicillin was such a wonder drug, we realized very quickly that we needed different types of antibiotics. So we knew that additional types, different methods of killing bacteria were needed. So we came up with some other types of antibiotics that were actually able to kill gram-negative bacteria. And one example of those is streptomycin. So streptomycin actually interferes with the protein synthesis process. So it doesn't matter that it can't reach that cell wall, because what it does is it actually is able to come in here and disrupt, you see this antibiotic here on this ribosome, it disrupts the process of protein synthesis, making it unable to produce proteins. But it does not affect eukaryotic cells because our ribosomes are different. And that antibiotic does not fit the human ribosome, and humans are able to make a functional protein. So both eukaryotic cells and prokaryotic cells have ribosomes, but our ribosomes are different sizes and different structures. So streptomycin is able to get in on these bacterial ribosomes and keep those proteins from being produced, making it so that that bacteria cannot live anymore. So another type of antibiotic that is able to target our gram-negative bacteria. We're also able to use streptomycin for gram-positive bacteria as well. And that concludes our lecture on chapter number 3, where we learned all about our cells. Please make sure, if you haven't already, that you read chapter 3 on Achieve, and then complete the chapter 3 adaptive quiz as well. And then kind of to go along with chapter 3, we also have class activity number 4 to complete too. So that is all for today's lecture. Mahalo for watching.